Okay, let's have the mitochondria, peroxisomes, and the chloroplast. I think the peroxisomes will be discussed tomorrow kasi mitochondria is quite longer and partly put ang chloroplast. So this is more on chapter 13. Now mitochondria, we know mitochondria is the powerhouse of the cell because it provides or produces a lot of ATP. It produces a lot of ATP than any other else. And aside from that, mitochondria also carries its own genome. It has its own genome compared to other organelles like lysosomes and endoplasmic reticulum don't have this kind of characteristics. So it has its own genome. And previously, mitochondria were known to have a resemblance with the bacteria, as this was discussed in last reading, that mitochondria derive from early bacteria or the aerobic bacteria engulfed by an archaea. And the archaea and the aerobic bacteria have showed a symbiotic relationship in which the aerobic bacteria provides energy for the archaea, and the archaea provide food for the aerobic bacteria to synthesize on. So, yun ang history ng endosymbiotic theory. So that is the story of the endosymbiotic theory. Now, the objective of this day that we have to illustrate the functional organization of mitochondria, describe mitochondrial genome, summarize how proteins and lipids are imported to the mitochondria, and explain the role of proton gradient to transport across the mitochondrial membrane. So first, let's discuss on the structure of the mitochondria. Mitochondria is one of the double-bonded organelle. So double-bonded or double-membraned organelle. So it has an outer mitochondrial membrane, and we have the intermembrane or the inner mitochondrial membrane. So IMM, kung outer is OMM. Later on, you will see some acronym OMM that means outer mitochondrial membrane. If it is IMM, that is the inner mitochondrial membrane. So it plays a critical role in production of ATP. So it performs a lot in metabolic during metabolism. Now, once the pyruvate once the glucose is broken down and transformed into a pyruvate by the process of glycolysis, if you could remember what is glycolysis, glycolysis happens outside the mitochondria, or it happens inside the cytoplasm or in the cytosol, and converting the glucose into a 3-carbon carbohydrate called the pyruvate. So the pyruvate will enter to the mitochondria and will undergo a metabolic pathway called the susunod na metabolic pathway to convert the Krebs cycle. So TCA, tricarboxylic acid cycle. So mangyari ang tricarboxylic acid cycle inside the matrix of the mitochondria. And later on, the energy will be again transported to the electron transport chain happen in sa intermembrane ng mitochondria. So they contain also their own DNA, which encodes tRNA, rRNA, and some mitochondrial proteins. While OMM is mostly permeable to ions and large molecules, but the IMM or the inner membrane, mitochondrial membrane is less permeable compared to OMM. So we have here the matrix, the space of the mitochondria, And these are the enfoldings of the inner membrane, intermembrane of the mitochondria, and this is the outer membrane of the mitochondria. So the enfoldings are called the cristae. So the enfoldings of the membrane forms stochastic. So this is the matrix where the PCA happens. So we have the citric acid or tricarboxylic acid or Krebs cycle, in which when the pyruvate was the glucose is converted to pyruvate, the pyruvate will enter the outer membrane passing through a pore. So merong pore ang inner mitochondrial membrane because it doesn't allow some of the molecules to enter. So the pyruvate will enter sa pore. Then in the matrix, it will combined with the acetyl-CoA, will have the citric acid cycle. Now the citric acid cycle produces NADH, and NADH, FADH2 will go to the electron transport chain or the ETC to produce more ATP. So this is how the oxidative metabolism in the mitochondria. So the inner mitochondrial membrane represents the principal site of ATP generation. So you have to remember that the inner mitochondrial membrane is the principal site for ATP generation. And the outer mitochondrial membrane is highly permeable to small molecules, but the inner mitochondrial membrane is lesser permeable compared to the outer. So the pyruvate will pass through to a pore called porins. So this is made up of protein. Now the mitochondria, aside from production of ATP, it also involves fusion, fission, and napato isang method na it was not included is mitophagy. Now, mitochondria is a dynamic organelle in which it also fuses with other mitochondria. So that is fusion. So, but in fusion of mitochondria, it requires some receptor sites. We call the MFN1 and MFN2 and the OPA1. So when you have these sites, it allows the mitochondrial membrane to fuse into one mitochondria. But aside from that, we have also the fission in which the mitochondria will divide itself. So munang in a story sa endosymbiotic theory, bakit ang mitochondria resembled or behaved like a bacteria? Because of this dynamic movement, the fission and fusion, in which it is also tells us that this is a simple binary fusion, which is a common behavior on a bacteria. And since mitochondria is very small, so that is another evidence. So third, mitochondria carries a genetic or a genome or a DNA. So yun yung three evidences that the mitochondria is related to bacteria. Now, so in fusion, it also requires some components like MID51, PIS-S1, MFF, DRP1, and MID49 and ER. So the fission begins when the endoplasmic reticulum is recruited to the constriction site. So this is marked by mitochondrial DNA. So that is one of the next multiple outer mitochondrial membrane bound proteins, so such as FIS1, MFF, MID49 and MID51. So ito yung mga coded genes na nasa mitochondria, and to recruit drop one to the surface of the mitochondria, adding ER-mediated membrane. So this is one way also to reproduce another mitochondria. But this kind of activity, ang fusion, mag-decrease lang ito when age comes. And mo ni ang crucial if the mitochondria will no longer divide, then one factor is aging or merong mga deficiency. So this is very important because mitochondria produces ATP and this will be used up by the cell. If the mitochondria will not multiply, then the cell will have a hard time to maintain its activity. Now, we have different fusion or fission proteins in different species. For example, in mammalian, the fusion proteins required, we have the MFN1, MFN2, OPA1, while in C. elegans, they have only two, identified two, which are FZO1, E3, and sa Drosophila melanogaster, we have FZO, MARF, and OPAL. So imagine kahit malaki ang C. elegans compared to the melanogaster, gamay lang siya o fusion proteins. While the fission proteins, in mammalian, we have here 1, 2, 3, 4, 5, 6. 6 identified proteins for fission. Okay, fission means these are the proteins used to separate or divide mitochondria from 1 into 2 organelles. So we have DRP1, FIS1, MFF, MID49, MID51, and DNM2. Now, to know the meaning of these words, it was present already sa inyong textbook. Katong gihatag na ko na PDF file na libro. So, yun ang source ng ano. Now, for the C. elegans, they have identified three proteins. DRP1, FS1, and FIS2. While in melanogaster, they have three. DRP1, FIS, DMEL. Now, the common thing of every three species, we have the DRP1 as a common protein for fission. Now, regarding with genetic system of the mitochondria, it said that it contains a genome, but not as many found in the nucleus. Konti lang, hindi siya gaano karami compared sa found in the nucleus. So, mitochondria are thought to have evolved from the bacteria. So, ito yung usually gina-discuss in the endosymbiotic theory, or we call it endosymbiosis, in which the archaea again engulf an aerobic bacteria, and instead of digesting the aerobic bacteria, it come up to have an symbiotic relationship in which aerobic bacteria provides energy for the archaea, and the archaea will provide food for the aerobic bacteria to break down the particles and utilize it in the metabolism. The mitochondrial genome are usually circular, like what we have in bacteria. So this is another evidence that the mitochondria had a resemblance to the bacteria because most of the bacteria have circular DNA. Sa eukaryotes, diba, we have the linear, helical, double helical DNA. While in mitochondria, it is circular na DNA. Ito singular din ito. So, para talaga siyang bacteria. So, it has a close relationship with the bacteria. And which are present in multiple copies per organelle. So mitochondrial genome encodes all the ribosomal RNA. So this is where the RNA came from. So dito nanggaling si RNA or yung ribosomal RNA, transfer RNA, and mRNA. So needed for translation of this protein coding sequence within the mitochondria. And the mitochondria have 13 proteins involved in the electron transport and oxidative phosphorylation. So electron transport happens in the intermembrane of the mitochondria. So sa inner membrane ng mitochondria. Now, mitochondrial DNA can be altered by mutation, like in bacteria also. Bacterial genome could be easily altered. Dali lang mag-mutate. That's why we have these mga inherited defective genes. Pwede siya ang murag diri ni gikan ang mga defective genes based on their theory. Since this is the genome that could be easily mutated. So, pwede siya ma-inherit sa next generation, yung defective gene ng mitochondrial DNA. So, since almost all the mitochondria of the fertilized eggs are contributed by oocyte rather than sperm. So, muni ang ginatawag nila na term na maternal DNA. So, the mitochondria carries the maternal DNA. It comes from the mother. So, less lang sa sperm. So, the germline mutations of mitochondrial DNA are transmitted to the next generation by the mother. So, one good example is the Leber's hereditary optic neuropathy. So, this is a disease that leads to blindness and can be caused by mutations of mitochondrial genes that encoded components of the electron transport chain. So, murag kumbaga, the mother, pwede niya i-pass on since kung ang mother na genetic defect or carrying a defect on mitochondria, pwede niya mapasa sa iyang anak. So this is another source of genetic diseases. Pwede mitochondrial, pwede punta sa nuclear na aspeto. Now, how the proteins are being transported to the mitochondria. So the mitochondria will import the proteins with pre-sequence. So once the proteins are released in the cytoplasm, and once the Golgi apparatus, excuse me. Kadali lahapos for a while. Ay, naku. Okay, so let's go back. Lumabas na ang bata sa kwarto. Wala akong may bantay. So, ito. So, this is a protein. So, kailangan, once the paano pumasok ang protein sa mitochondria? It requires the HSP70. So, para siyang facilitated diffusion. It requires another protein to guide the pre-sequence protein passing through the outer membrane, then since mas selectively permeable ang inner membrane, then it requires mga different protein transporters. Okay, so we have here the pores. We have the TOM in the outer membrane. We have the TOM complex. And the inner membrane, there's another pore called the TIM23 complex. Now, sa systolic, sa outer membrane, the protein requires ATP. It uses an energy. And at the same time, a protein called HSP70. So this is the pre-sequence protein. Papasok siya. So once it is activated, papasok siya sa TOM complex in the outer mitochondrial membrane. Now, once it passes through sa TOM complex, it enters now to the TIM. So some proteins with transmembrane domain exit to the TIM23. So dito siya mag-exit sa TIM23 complex. Now, the proteins destined for the mitochondrial matrix associate with soluble HSP70. So another protein will meet the pre-sequence protein and attach again the HSP70 para i-coil niya into a globular protein. Then it uses an energy. So everything will be uses an energy. So MPP is another protein complex that will import HSP70 attached to the pre-sequence protein. Next, protein targeting to the mitochondrial inner membrane. So this is another path. The same thing, it passes through from the cytosol. The protein requires need to be attached with HSP70. Without this one, hindi pwede makapasok ang pre-sequence protein. So, kailangan ng isang transporter in order to pass through the TOM complex and the TIM22 complex. Okay. So, this is a similar pathway lang. So that's how the protein enters the mitochondrial membrane. So it requires the ATP or energy in order to connect the HSP70. And also, since the membrane is too tight, it requires the protein, integral proteins to as a gateway for the pre-sequence protein to enter the mitochondria. So not only the proteins, but also the lipids is imported from the cytosol. So lipids like in animals, we have the sphingolipids, cholesterol, phosphatidylcholine, phosphatidyllinositol, and phosphatidylserine are also synthesized in a smooth endoplasmic reticulum, could be also transported to the mitochondria. So hindi lang protein ang pinapasok ni mitochondria, but also the lipid components. Synthesized by the ER, specifically sa smooth, because si rough ER, protein ang kanyang ginasynthesized, while the smooth endoplasmic reticulum are more on lipids. So these are examples of lipids. So if you're going to recall again, yung specific function ng ER, so we have there 6. Then kung specific function ng smooth ER, primary function ng smooth ER is the synthesis of the lipids. So isa yun sa mga function ng smooth. Now, the mitochondria then synthesize phosphatidyletanolamine formed from the phosphatidylserine. And mitochondria also catalyze the synthesis of the unusual phospholipid cardiolipin. Now, what is the importance of cardiolipin? Cardiolipin is a localized inner membrane of mitochondria. So it is found in the inner membrane mitochondria, which act as an important sa oxidative phosphorylation during electron transport chain. So, importante si cardiolipin. So, without these lipids, kung hindi ito siya masynthesized, then energy production in electron transport system would not be possible. And ma-affected na din ang TCA because TCA also uses FAD, NAD. So, para mahimong active a type of energy. So, how the lipids are transported in the membrane? Now, from ER, so from the phospholipid transfer protein, extract phospholipid molecules from the endoplasmic reticulum and transport them to the mitochondria on the outer membrane. So we have another type of protein. Now, from the ER, endoplasmic reticulum, it will get the phospholipid from the membrane. And this protein will pick the molecule and transfer it to the mitochondrial outer membrane. Amazing, no? Wala niya siya na-discuss sa other books. But makita niya na how the proteins, importante ang proteins. So proteins are very important, not as part sa enzyme to hasten chemical reactions, but also proteins are used as parang transporters ng molecules, other molecules. Like this one, the lipids, the phospholipid molecule was from the ER going to the mitochondrial outer membrane. Imagine mo parang robot. So the transport of metabolites across the mitochondrial inner membrane. So what are these metabolites? So we have the OH, we have, now the pH is quite different sa intermembrane spaces. So we have the, in the mitochondria, since we have outside the cytosol, this is the OMM, the outer membrane. This is the intermembrane space. So this is the space between the OMM and IMM. So this layer is the IMM. Now, in the matrix, the pH is basic, is more basic, which it has a pH of 8, while sa intermembrane spaces, it is neutral. The pyruvate could enter the integral proteins. So these are all proteins, this one. So could be, they could enter through electric potential. So it requires energy. And also could be transported through concentration gradient. So kung higher concentration, some of the molecules could enter from higher concentration to a lower concentration. Now, mitochondria was discovered by Margit Nas and Sylvian Nas. Margit M.K. Nas and Sylvian Nas. I don't know kung mag-asawa ito or magkapatid. And the DNA, microscopy of DNA sensitive thread inside the mitochondria was seen by Ellen Hasselbronner, Hans Stupi, and Gottfried Schatz. So they do the biochemical assay on mitochondrial fractions. So the difference between, what is the difference between the mitochondrial DNA and the nuclear DNA? First, mitochondrial DNA is circular like what we have in bacteria, while the nuclear DNA in eukaryotes is linear and helical. Both are double-stranded. Both are double-stranded. So, yun lang ang difference. Mali yung sinabi ko kanina kasi single yung na-mention ko last time. So, both are double-stranded. The mitochondrial DNA is uniparentally inherited. So, we inherit this sa ating mother. So, mitochondrial DNA is passed from mother to offspring, because it was found in the egg, not merely on the sperm. Nuclear DNA is diploid, whereas mitochondrial DNA exists in a state of ploidy. So it contains numerous mitochondria, mas heteroplasmic state, so mas marami, hindi siya more than the diploid. Now, let's proceed sa chloroplast. Chloroplast could be found only in plants, but not in animals. So, this is the unique organelle found in plants. And like mitochondria, chloroplast has its own genetic genome. So, like also the mitochondria, chloroplast could produce, it is also the site of photosynthesis, and it comes in different forms. So depending sa type of plant. We have the amyloplast, which could store starch, chromoplast, store pigments for fruits and flowers. Chloroplast, store chlorophyll, function in photosynthesis, and found in leaves and other plant organs. Pwede siya sa stem, but not all plants have brown stem. So sa mga shrubs, green stems, pwede pa. Pero kung mag-brown, it was already covered with cork and some bark. So wala na yan siyang chloroplast. So based on the structures of chloroplast, so we have one chloroplast. Like the mitochondria and the other organelles, it consists of two membranes. We have the outer membrane, the green one, marag schematic diagram lang to show kung ano ang itsura. Then the inner one, we have the inner membrane represented by an orange layer. What's inside the chloroplast? We have the stacks of thylakoids. So mura siya coins. Kung yato ni ma-remember, thylakoids are like coins, stack of coins. And the space is called the aqueous, called the stroma. So this is the aqueous stroma. So parang may fluid in between when we say aqueous. So a group of thylakoids is called granum. So granum, a group of thylakoids. So internal fluid space, we have the stroma and DNA. Where could be found the DNA, the thylakoids, and the grana. So chloroplast function is more on photosynthesis in plants. So it generates ATP and synthesizes sugar like what we have sa mitochondria. So it transforms the solar energy into a chemical energy. Ang sa mitochondria lang kasi is transforming the chemical energy to another form of chemical energy. From our organic molecule to a chemical compound in the form of energy. So it is semi-autonomous. Like some mitochondria, it also undergo binary fission in development. It also carries a DNA, like the mitochondria. So it divides like a bacteria through binary or multiple fission. Now, chloroplasts contain an internal thylakoid membrane system. So, muna yung interthylakoid, katong stacks of thylakoid, chorograna. So, the thylakoid membrane forms a network of flattened disc. Paggrupo na grana. So, in particular, with the three membranes, divide the chloroplast into three distinct internal compartments. Okay, first, we have the intermembrane space. So, this is a space between the outer membrane and the surface of the inner membrane. So this is the intermembrane space. Next, the stroma, which lies inside the envelope. So this is the stroma, the aqueous space, so filled with the fluid, and the thylakoid lumen. So if we're going to cut the thylakoid, you will see the space in between the thylakoid. So that is the thylakoid lumen. The chloroplast genome consists of 150 genes. So chloroplast genomes also encode approximately 50 proteins. So these are some of the proteins in the plants coming from a chloroplast. We have the gene expression for rRNA, tRNA, ribosomal proteins, RNA polymerase subunits. In the photosynthesis naman, we have the photosystem 1, photosystem 2, the cytochrome VF complex, ATP synthase, and ADP reductase. We have the metabolism. We have 11 genes coded for that, and protein folding and membrane insertion. We have 14 genes. Like sa mitochondria, it also import proteins. Para naman pang ano. So sa mitochondria, di ba, we have katong complexes. Yung team 23. So plants also has its own complexes. So the TOC complex. So para siyang pore, like what we have in mitochondria. So the import of proteins into the chloroplast stroma. So proteins with N-terminal transits, peptides, and are targeted to the TOC complex or the TOC complex. So it requires again an energy. It also requires a protein called HSP. So HSP is very important component in order to guide the peptide going to the stroma. So this protein, thread of protein, cannot enter immediately because it requires the HP para i-activate niya ang TOC complex, para ma-open ang TOC complex. Then that's the time this thread of proteins could enter the outer membrane of the chloroplast. Then passing through the intermembrane, it requires another protein to receive this thread. So HSP93 will bind to the thread of proteins para magiging HS70. So like lang sa pareho lang sila ng process sa mitochondria. It requires again energy. So import of proteins into the thylakoid lumen. So we have the TAT pathway, the SEC pathway, and the SRP, hindi siya suggested retail price. It is another type of protein. It is the signal recognition particle. So this SRP is a signal recognition particle. Ang SEC pathway, so we have the TAT pathway. The TAT pathway, a thylakoid signal sequence containing 2 arginines near the amino terminus target. So, kailangan may arginine as the terminus. So, this is the terminus, the terminal end of the protein. So, para ma-recognize siya sa protein complex, para makapasok siya. So, what is TPP? That is the thylakoid processing protease. So, this is an enzyme. So, the enzyme will cut the arginine terminal end and the enfolding of proteins inside the thylakoid lumen. Now, another path naman, we have the SEC pathway. Okay, what is the SEC pathway? This is another pathway. So, the SEC-A, so this is the SEC-A. It is a protein also that recognizes the thylakoid signal sequence and targets the protein to the SEC translocon. So, kailangan may SEC translocon. So, this is HSP70. Using energy derived from ATP hydrolysis to transfer the protein to the lumen. So, murag iya, kumbaga, si SEC-A is a protein that will identify the protein, but kailangan yung protein thread, it has the AHP70 para mapasok niya. So, pero itong HSP70 will live on the lumen. It only allow this thread of proteins passing through the lumen, the pore. So, what's next? Some transmembrane proteins exit the SEC translocase laterally for incorporation in the thylakoid membrane. So, some of the proteins may also enter sa thylakoid lumen or dito sa intermembrane. So, pwede siya ma-embed within the membrane. Some will go to the thylakoid lumen. And we have the TPP also. The TPP will cut off the terminal end. So, it will cut on the terminal end containing the SEC translocon. Next, we have the third one, the suggested retail price, no? That's the signal recognition particle. Okay, the signal recognition particle, it requires the mRNA and the ribosome. So that is more on the translation. So after translating the protein sequence, releasing a protein. Now, we have the CPRS. This is the chloroplast signal recognition particle attaches to the newly transcribed protein. But it requires the terminal end for the CPSRP to attach with. Kung wala iyan, wala itong code na to, this protein could not attach to the protein sequence. So, kung wala itong code na to dito sa protein sequence, hindi mag-attach si CPSRP. So, for the CPSRP, it attaches itself to a certain end on the sequence. Kasi may recognition siya, na-recognize niya. Dikit si ang acronym, CPSRP. Then, it will lead to the the pore or the terminal or the channel ALV3. What is AL? This is enzyme translocase. So inserted into the thylakoid membrane by LLB translocase. So there's an enzyme. So the enzyme will insert the protein sequence in the thylakoid membrane. So in RSP pathway, the protein will be guided and inserted in the membrane. While in SEC pathway, could either be the protein will be accompanied by certain protein passing through a channel, then the protein could either be going to the thylakoid lumen or in the membrane. While in that pathway, so the protein sequence and the newly sequenced proteins will enter the channel, passing through the channel and stayed at the lumen. Hindi siya ma-inserted sa intermembrane. So nakita nyo ang difference between the TAT pathway, the SEC pathway, and the SRP pathway. But ang kailangan? Kailangan meron tong terminal end to allow some proteins to attach, to lead the way para papunta sa chloroplast. Kung wala yan, hindi siya para sa kanya. Hindi para sa kanya yung protein. Baka sa ibang organelle. So, munang identifying mark ng other organelles that this protein, newly transcribed protein, is for them. So, kailangan may mga recognition terminal ends. Now, in plastids, we have different plastids in plants. We have the ethioplast, chromoplast, chloroplast, leucoplast, amyloplast, elioplast, and proteinoplast. Excuse me. Now, ethioplast, these are crystalline prolamellar bodies, precursor of chlorophyll. Chromoplast, this will show pigments and synthesize pigments and storage. Chloroplast, ang chromo, pigment synthesis, chloro, photosynthesis, and lipid synthesis. Leucoplast, synthesis of fatty acids and amino acids. Amyloplast, storage of starch and sugar. Elioplast, storage of lipids and oils. Kung natay mga plants na naay mga oils and rich in oils, meaning they have this kind, they have a rich elioplast, because there are some also plants that contains less oil. Anong dira nato ginakuha itong mga essential oils ng mga plants. And we have proteinoplast, site of enzyme for protein activity. So these are the different kinds and types of plastids. So that's the end of then tomorrow we shall